Hello, everyone, and welcome to today's keynote presentation, Unraveling the Role of the AP4 Adaptor Complex in Protein Sorting and Neurological Disease. This webinar is part of the Cell Biology virtual event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational webinar is presented by LabRoots. So let's get started. Before we begin, I want to remind you that this event is interactive, and we encourage you to participate by first communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you're having any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that Continuing Education Credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Juan Bonifacino, Associate Scientific Director of Neurosciences and Cellular Structural Biology in the division at Eunice Kennedy Shriver National Institute of Child Health and Human Development and the National Institute of Health. Dr. Bonifacino was elected for delivering the keynote address of the American Society for Cell Biology today. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Bonifacino, welcome. You may now begin your presentation, sir.
Thank you very much, Susie, for the introduction. And I'm very pleased to be here this morning. And good morning, everyone, or good afternoon, wherever you are today. So as Susie said, I'm an investigator at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. And my lab does mainly basic research on molecular mechanisms of protein trafficking, how proteins get to different compartments within the cell. But we also apply knowledge gained from those basic studies to the understanding of molecular mechanisms of disease, in particular neurodevelopmental disorders. And today I would like to present an example of that translation from basic research to disease-related research. As Susie mentioned, I will be talking about the AP4 adaptor complex. It's a component of the machinery that ensures that certain proteins are properly delivered to their respective compartments within the cell, and how defects in this complex cause a particular disease known as hereditary spastic paraplegia. So the eukaryotic cell comprises a number of membrane-bound compartments, some of which you will be able to appreciate in this electron micrograph of a pituitary cell. In particular, this is a cell that makes growth hormone and therefore has growth hormone containing granules, which you will be able to appreciate in the interior of this cell. And you will also be able to see a number of other membrane-bound compartments, including the nucleus in the center of the cell, the plasma membrane that surrounds the cell, and other organelles such as the endoplasmic reticulum, the Golgi complex, endosomes, lysosomes, and others. Each of these intracellular compartments has a different protein composition, which is responsible for the specific functions of those organelles. And what we and other people in this field are interested in understanding is how is that heterogeneous distribution of proteins within the cell achieved? In other words, how do some proteins go to the nucleus, whereas others go to the plasma membrane, or to endosomes or lysosomes? What molecular mechanisms ensure that these proteins go to the correct compartments within the cell? So that's the problem that we are interested in. And in particular, we are interested in organelles that make up the so-called endomembrane system of the cell, which comprises the endoplasmic reticulum on the left, the Golgi complex in the center, various types of endosomes and lysosomes, and different domains of the plasma membrane. There are many different machineries that ensure that proteins are able to navigate this endomembrane system and be delivered to their respective compartments within the cell. Among those components of the machinery are four so-called adaptor protein complexes, or AP complexes: AP1, AP2, AP3, AP4, and AP5, which are shown on this scheme at the locations where we think they act. And what these complexes do is they select specific proteins that we call cargos for incorporation into transport vesicles that then will move from one compartment to the other. These adaptor protein complexes are composed of four subunits, which are shown here in schematic fashion in different colors. They bind to membranes through interactions with specific membrane phospholipids and specific GTPases. So these are membrane-bound complexes. And then they function to recognize sorting signals in the cytosolic tails of transmembrane proteins, leading to the concentration of those transmembrane proteins within areas of the membrane that contain these adaptor protein complexes. In other words, what these AP complexes do is grab transmembrane proteins by the tail and concentrate them in regions of the membrane that would give rise to a transport vesicle. Then the adapters recruit scaffolding proteins such as Clathrin, which is the best known of these scaffolding proteins, leading to the formation of a protein coat on the membranes that by electron microscopy can be seen as an electron-dense deposit on the plasma membrane, as shown here in this electron micrograph of a so-called Clathrin-coated pit at the plasma membrane. So these Clathrin-coated areas of the membrane concentrate cargo proteins via interactions with the adaptor protein complexes and away from other proteins that don't have those sorting signals and that remain at the plasma membrane. These Clathrin-coated pits then invaginate, give rise to a coated vesicle, which then delivers those cargoes, those proteins, to another compartment within the cell. In the particular case of plasma membrane proteins, they are internalized into so-called endosomes. Similar processes take place not only at the plasma membrane, mediated by the AP2 complex, but also the trans-Golgi network and various endosomal compartments through the function of complexes such as AP1, AP2, AP3, AP4, and AP5. In essence, what all of these complexes do is they recognize specific transmembrane proteins via recognition of sorting signals in the tails of these proteins. That leads to the capture of those cargoes within coated vesicles that then deliver the vesicles from one compartment to another. Now, these 5 complexes, AP1 through 5, have similar but also different proteins. They are recruited to different membranes via interactions with different membrane phospholipids or different GTPases. And I'm not going to get into that detail. But for this talk, it's important to note that mutations in subunits of each of these 5 complexes have now been reported to cause different genetic diseases, different genetic disorders, which are shown here. And I'm
Hello, everyone, and welcome to today's keynote presentation, Unraveling the Role of the AP4 Adaptor Complex in Protein Sorting and Neurological Disease. This webinar is part of the Cell Biology virtual event, and I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots. So let's get started. Before we begin, I want to remind you that this event is interactive, and we encourage you to participate by first communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the right of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. Now, if you're having any trouble seeing or hearing this presentation, simply click on the Help Desk button located at the bottom of your screen within the navigation bar or from the lobby. As a reminder, this presentation is educational and offers free continuing education credits. Click on that Continuing Education Credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Juan Bonifacino, Associate Scientific Director of Neurosciences and Cellular Structural Biology in the division at Eunice Kennedy Shriver National Institute of Child Health and Human Development and the National Institute of Health. Dr. Bonifacino was elected for delivering the keynote address of the American Society for Cell Biology today. For a complete biography of our speaker, please visit the Biography tab at the left of your screen. Dr. Bonifacino, welcome. You may now begin your presentation, sir.
Thank you very much, Susie, for the introduction. And I'm very pleased to be here this morning. And good morning, everyone, or good afternoon, wherever you are today. So as Susie said, I'm an investigator at the Eunice Kennedy Shriver National Institute of Child Health and Human Development. And my lab does mainly basic research on molecular mechanisms of protein trafficking, how proteins get to different compartments within the cell. But we also apply knowledge gained from those basic studies to the understanding of molecular mechanisms of disease, in particular neurodevelopmental disorders. And today I would like to present an example of that translation from basic research to disease-related research. As Susie mentioned, I will be talking about the AP4 adaptor complex. It's a component of the machinery that ensures that certain proteins are properly delivered to their respective compartments within the cell, and how defects in this complex cause a particular disease known as hereditary spastic paraplegia. So the eukaryotic cell comprises a number of membrane-bound compartments, some of which you will be able to appreciate in this electron micrograph of a pituitary cell. In particular, this is a cell that makes growth hormone and therefore has growth hormone containing granules, which you will be able to appreciate in the interior of this cell. And you will also be able to see a number of other membrane-bound compartments, including the nucleus in the center of the cell, the plasma membrane that surrounds the cell, and other organelles such as the endoplasmic reticulum, the Golgi complex, endosomes, lysosomes, and others. Each of these intracellular compartments is responsible for the specific functions of those organelles. And what we and other people in this field are interested in understanding is how is that heterogeneous distribution of proteins within the cell achieved? In other words, how do some proteins go to the nucleus, whereas others go to the plasma membrane, or to endosomes or lysosomes? What molecular mechanisms ensure that these proteins go to the correct compartments within the cell? So that's the problem that we are interested in. And in particular, we are interested in organelles that make up the so-called endomembrane system of the cell, which comprises the endoplasmic reticulum on the left, the Golgi complex in the center, various types of endosomes and lysosomes, and different domains of the plasma membrane. There are many different machineries that ensure that proteins are able to navigate this endomembrane system and be delivered to their respective compartments within the cell. Among those components of the machinery are four so-called adaptor protein complexes, or AP complexes. AP1, AP2, AP3, AP4, and AP5, which are shown on this scheme at the locations where we think they act. And what these complexes do is they select specific proteins that we call cargoes for incorporation into transport vesicles that then will move from one compartment to the other. These adaptor protein complexes are composed of four subunits, which are shown here in schematic fashion in different colors. They bind to membranes through interactions with specific membrane phospholipids and specific GTPases. So these are membrane-bound complexes. And then they function to recognize sorting signals in the cytosolic tails of transmembrane proteins, leading to the concentration of those transmembrane proteins within areas of the membrane that contain these adaptor protein complexes. In other words, what these AP complexes do is grab transmembrane proteins by the tail and concentrate them in regions of the membrane that would give rise to a transport vesicle. Then the adapters recruit scaffolding proteins, such as Clathrin, which is the best known of these scaffolding protein, leading to the formation of a protein coat on the membranes that by electron microscopy can be seen as an electron-dense deposit on the plasma membrane, as shown here in this electron micrograph of a so-called Clathrin-coated pit at the plasma membrane. So these Clathrin-coated areas of the membrane concentrate cargo proteins via interactions with the adaptor protein complexes and away from other proteins that don't have those sorting signals and that remain at the plasma membrane. These Clathrin-coated pits then invaginate, give rise to a coated vesicle, which then delivers those cargoes, those proteins, to another compartment within the cell. In the particular case of plasma membrane proteins, they are internalized into so-called endosomes. Similar processes take place not only at the plasma membrane, mediated by the AP2 complex, but also at the trans-Golgi network and various endosomal compartments through the function of complexes such as AP1, AP2, AP3, AP4, and AP5. In essence, what all of these complexes do is they recognize specific transmembrane proteins via recognition of sorting signals in the tails of these proteins. That leads to the capture of those cargoes within coated vesicles that then deliver the vesicles from one compartment to another. Now, these 5 complexes, AP1 through 5, have similar but also different proteins. They are recruited to different membranes via interactions with different membrane phospholipids or different GTPases. And I'm not going to get into that detail. But for this talk, it's important to note that mutations in subunits of each of these 5 complexes have now been reported to cause different genetic diseases, different genetic disorders, which are shown here. And I'm not going to get into most of these genetic disorders, and we collectively refer to them as coatopathies because they are diseases of protein coats, and defects in subunits of these complexes lead to missorting, mislocalization of different cargoes, and that leads to different diseases. In today's talk, I am going to talk about coatopathies caused by mutations in subunits of the AP4 complex, which are known as hereditary spastic paraplegia, or HSP, or also abbreviated SPG. These are synonyms, essentially. Types 47, 50, 51, and 52, which are caused by mutations in each of the 4 subunits of the AP4 complex. Now, let me tell you a little bit about the AP4 complex. I refer to this complex as the poor cousin of the AP4 complex family because for many years there was very little interest in this complex. It has 4 subunits named epsilon, beta 4, mu 4, and sigma 4. It was discovered in 1999 in my lab and also the lab of Scotty Robinson at the University of Cambridge in the UK. And as you can see in that gel, those are the 4 subunits of the complex. That's how we first discovered it, biochemically. We knew that there was this complex, but we didn't know what it did. So a number of biochemical and cellular analysis then showed, first of all, that this complex is evolutionarily ancient. It's present not only in humans, but also in other eukaryotes, including a lot of protozoa, for instance. It's also ubiquitous. It's found in every cell in our body. Now, this complex, however, is present at very low levels. And this made the study of the localization and function of this complex quite difficult. And that's one of the reasons why for many years there was little interest in this complex, simply because it was very difficult to study. It's part of a non-Clathrin coat, unlike the AP2 complex, which localizes to the plasma membrane. It's associated with the trans-Golgi network, which is the subcompartment of the Golgi complex. As you can see on the immunofluorescence microscopy below, the arrows are pointing to the trans-Golgi network. And so that's where AP4 is located. So it's probably playing a role in that organelle, in that compartment. It's recruited to the membrane by small GTPases of the R family. And the mu4 subunit, which is depicted in blue in the scheme, binds sorting signals in the tails of in the cytosolic tail of transmembrane proteins, fitting a particular consensus motif. And the most important thing to note here is that those signals have a critical tyrosine or Y residue. That's what the mu4 subunit of AP4 recognizes in the tail of transmembrane proteins. And we even know the structure of the mu4 subunit of AP4 bound to a sorting signal depicted in blue and yellow, respectively. Mu4 is in blue, and the sorting signal is in yellow. And so all of these studies revealed a lot of properties of the AP4 complex. We knew where it was in the cell, how it was recruited to membranes, what kind of sorting signals it recognized, and how it recognized it in exquisite detail, even atomic level resolution detail. However, despite of all of these studies, we did not know what the physiological role of AP4 was for a long time, for almost 15 or 20 years. And a breakthrough occurred when several groups demonstrated that mutations in each of the 4 subunits of AP4 cause a human disease known as hereditary spastic paraplegia, or HSP. Type 47 caused by mutations in the beta-4 subunit, 50 caused by mutations in the mu4 subunit, 51 caused by mutations in epsilon, and 52 caused by mutations in sigma-4. Irrespective of which subunit is mutated, the patients have the same disease, because mutation in one subunit results in an inability to assemble the AP4 complex and in similar cellular defects. So what is this disease, hereditary spastic paraplegia? Well, it's characterized by stiffness and weakness of the legs. So it's essentially similar to what's known as cerebral palsy. The patients are children, and they have difficulty walking. And this is caused by axonopathy of corticospinal tracts. In essence, the axons of neurons that are located in the cerebral cortex and that run all the way to the spinal cord are abnormal. They degenerate, and that caused problems with walking. Now, defects in AP4, mutations in AP4, cause a form of hereditary spastic paraplegia. And as you can see in the pictures on the left, the children need assistance for walking. They need walkers. And but in addition, these patients with mutations in AP4 have other problems, including intellectual disability, seizures, microcephaly, and it evolves to tetraplegia, in which they have problems not only with their legs, but also with their hands. So AP4 deficiency is a complicated type of hereditary spastic paraplegia in which, in addition to the movement disorder, there are a number of other neurological problems. And neuroimaging analysis of these patients show that they have a thin corpus callosum. The corpus callosum is the structure in the brain that connects both brain hemispheres. And in the case of these patients, it's very thin, meaning that there are fewer connections between the two hemispheres. And there is also evidence of axonal dystrophy, of damage to the axons in these patients. And here you will see a little video, which is available in YouTube, of the two first patients in the US who were diagnosed with this disease. And in particular, these are two girls, Molly and Robbie, who have mutations in the beta-4 subunit of AP4. And this is the day that they met for the first time, these two patients, at Boston Children's Hospital. And you can see that they have problems walking, and one of them is using a walker that particular day. This is their parents. And they were about 3 years old at this time. And as you can see, they have problems walking, but they still have use of their hands. They can play together. And let me see, was this all? Yeah, okay. And let me advance. And so those were the first two patients diagnosed in the US, but more than 200 cases have been reported in the literature throughout the world. And the belief is that this is probably, although it's a rare disease, it's probably more common than currently known, because it may be underdiagnosed as cerebral palsy, which up until recently was thought to be caused by trauma during birth. But nowadays it's believed to have also a genetic component. And in fact, some patients who had been initially diagnosed with cerebral palsy have recently been found to have mutations in AP4. So it may be more common than it's currently known. One problem with these patients is that the disease is progressive. It gets worse with age. You saw that those girls were still able to interact with each other and with their parents. They were still social. And they could speak a few words, but they will lose all of their abilities. Eventually, they will become tetraplegic. They will lose use of their hands, and they will be confined to a wheelchair because the disease is progressive. It has a neurodevelopmental component. The children are born with some problems, but it has also a neurodegenerative component, so it gets worse with age. So that's a problem. Essentially, the parents see their children wither away over time. But the progressive nature of the disease also offers an opportunity for early intervention. If we had some therapeutic intervention, we could use it early, and hopefully that would slow progression. The problem is that there is currently no treatment other than symptomatic treatment. And the problem is that, so these findings demonstrated that AP4 is certainly important for the development and function of the central nervous system. So when we started this work, we didn't know what AP4 did or what it was important for. These genetic studies demonstrated that it's important for the function of the central nervous system. So the problem is that we don't know what AP4 does at the cellular level, how AP4 deficiency causes hereditary spastic paraplegia. And this is something on which we concentrated our efforts for the past few years. And one approach that we took, let me silence this, was to develop a knockout mouse with a defect in one of the subunits of AP4, to use it as a model system for the human disease. And initially we were disappointed. This is the AP4 knockout mouse. We looked at it, and it didn't seem to have any problems. The mice were viable. They had no overt abnormalities. They had a normal lifespan, and they were even fertile. You could breed male and female mice with who were homozygous for AP4 knockout, and you could obtain a progeny. So we were initially concerned that maybe the AP4 knockout mouse was not recapitulating the problems with the human disease. However, we took our mice to a colleague of ours, Claire Lepichon, in our institute, who is a neurobiologist, a neuroscientist. And when she picked up the mice by the tail, she immediately realized that there was a neurological phenotype known as clasping. So when you pick a normal mouse, a wild-type mouse by the tail, the mouse will splay their hind limbs, probably in preparation for landing on a surface. So that's a spinal cord reflex that any mice will show. However, when Claire picked up the knockout mouse, she observed that the mice kept their hind limbs retracted. So they were unable to recognize the danger of falling on a surface without splaying their hind limbs. So this clasping phenotype is very common is a very common phenotype in neurological disorders in the mouse, and for the first time told us that the mice did have a neurological phenotype. We only had to challenge them in order to see those phenotypes. So we then performed other challenges, other neurological tests in these mice. One of them is called the rotarod test, in which you place the mouse on a rotating cylinder which rotates at increasing speeds, and then you measure the speed, the RPMs, at which the mice fall from the rotating rod. And as you can see here, the knockout mice fell at lower speeds than the wild-type mice, indicating that they did have a motor disorder. We also performed a so-called grip strength test, in which we place the mouse on a grid, and then we pull from the tail, and we measure the force at which they release the grip on the grid. And as you can see in the bar graph, the mutant mice also had reduced grip strength, showing that even their their limbs had reduced strength. And we also performed immunohistochemistry of the brain, and we saw in the lower picture that the knockout mice had a thinner corpus callosum relative to the wild-type mice in the upper picture, also similar to the human patients. And finally, we perform immunohistochemical staining of various parts of the central nervous system. In this case, it's the cerebellum. And you will notice in those pictures shown with the arrowheads that the knockout mice had swellings in the axons called spheroids that were absent in the wild-type cerebellum, indicating that these mice also had axonal abnormalities, axonal dystrophy. So in summary, the knockout mice had motor deficits, a thin corpus callosum, and axonal dystrophy similar to the human patients, indicating that these mice are a good animal model for the human disease, meaning that if we were able to develop some therapies, they could be first tested in the mouse before they are used in the humans. Okay, but we still have not answered the question, what is the cellular function of AP4? What does AP4 do in the cells so that its deficiency causes a neurological defect? And we thought that the key to answering this question was the cargos. Because what do adapter protein complexes do? They sort cargos. They grab cargos by the tail. They pack the cargos into transport vesicles so that they can be delivered to the right compartment. So AP4 must have some specific cargo that is missorted when we mutate AP4 in mice or when it's mutated in the patients. So we decided that if we were going to understand AP4 function, we needed to identify the cargos. And in order to do that, we performed what's known as an affinity purification of proteins that bind to AP4 and identification of those proteins by mass spectrometry. And I will not get into the details of those analyses. Suffice it to say that we identified a number of AP4 interacting proteins. These included the 4 subunits of AP4, which are highlighted at the top of this list: epsilon, beta 4, mu 4, sigma 4. The 4 subunits co-purified as expected. And then we identified a number of AP4 interacting proteins. And I would like to highlight here a protein known as ATG9A, which means autophagy-related protein 9A. It's a component of the cellular autophagy machinery, the machinery by which cells eat themselves. And I will talk a little bit more about that later in the talk. Okay, so AP4 recognizes ATG9. ATG9 is a transmembrane protein and therefore a putative cargo for AP4. It has 2 cytosolic tails, an N-terminal tail and a C-terminal tail, which are indicated by NH2 and COOH. So those are sequences that are facing the cytosol of the cells. And in particular, we noticed that the N-terminal tail, the NH2 tail, had a sequence YQRLE, which is highlighted in red, that fits the consensus motif for signals that bind to the mu4 subunit of AP4. So therefore, this was indeed a putative AP4 cargo protein. To demonstrate that, we used what's known as a yeast-to-hybrid system, which is used to demonstrate protein interactions, protein-protein interactions. So, and I will not even get into the details for how this works, but in this assay, when the yeast grows as colonies, it means that there is an interaction between the two proteins that are being tested. And in this particular test, we saw that ATG9A interacted with the mu4 subunit of AP4 as indicated by the colonies on the left side of the red box. There you can see that there is growth. That means that mu4 interacts with the tail of ATG9A. This is confirmation of the affinity purification and mass spectrometry analysis. And we also saw that when we mutated the tyrosine, the Y residue in the YQRLE sequence, that mutation abrogated the interactions, as you can see next to the colony in which there is growth. When we mutate the tyrosine to an alanine residue, there is no more growth, meaning that there is no more interaction. So this demonstrated that the mu4 subunit of AP4 recognizes the cytosolic tail of ATG9A via that tyrosine-containing sequence, as expected, demonstrated that indeed there is specific interaction of ATG9A with AP4. But what is the physiological importance of that interaction? In order to address this question, we looked at the distribution of ATG9A in wild type and knockout cells. In this particular case, the cells are mouse embryonic fibroblasts, or MEF, stained for endogenous ATG9A. Here you can see two cells, and in green you can see the distribution of ATG9A. You will notice that it's found throughout the cytoplasm, although it's more concentrated in the center of the cell, in the region where the Golgi complex and the trans-Golgi network are located. This is in wild-type cells. And then we looked in mouse embryonic fibroblasts with a knockout in the epsilon subunit of AP4. And this is what we saw. The results were really striking. I remember when the postdoctoral fellow called me to the microscope to show me the results, and these results really knocked my socks off because it was so striking. In the knockout cells that don't have AP4, ATG9A is concentrated in the Golgi complex and the trans-Golgi network and depleted from the periphery of the cell. So for the first time, we could see what AP4 was important for in cells. It was important to export ATG9A from the trans-Golgi network to the periphery of the cell. This is in mouse embryonic fibroblasts from the mouse that we had previously developed. What happens in humans? So we then looked in skin fibroblasts from a normal human individual, and it's the same as in the mouse embryonic fibroblasts. And then we looked in skin fibroblasts from a patient with a mutation in the mu4 subunit of AP4, and this is what we saw. Just like in the mouse fibroblasts, ATG9A was concentrated, was accumulated in the trans-Golgi network and depleted from the cell periphery, also in the human patients. Okay, so we didn't know what AP4 did. We now know that AP4 mediates export of ATG9A from the trans-Golgi network, or TGN, towards the periphery of the cell, as shown in this red box. And it shows ATG9A going to a PAS, which stands for pre-autophagic structure. ATG9A is an autophagy protein, and it functions in autophagy. And this is what I would like to tell you a little bit about, because we think that this is related to the pathogenesis of the disease. Okay, autophagy. What is this pathway? Autophagy means self-eating, cells eating themselves. So when do cells eat themselves? Well, under conditions of starvation, for instance, cells will eat some of their cytoplasmic components, mitochondria, endoplasmic reticulum, other organelles, in order to generate nutrients that they need to survive until nutrients become available again in the medium. But it's also a mechanism to dispose of foreign or damaged particles, including bacteria and viruses, protein aggregates that form in certain diseases, including many neurodegenerative disorders such as Huntington's disease, Alzheimer's disease, Parkinson's disease. And so the way it works is that these cytoplasmic particles are engulfed into a pre-autophagosomal structure that then closes into an autophagosome that you will see in the middle of this pathway that fuses with a lysosome, a degradative organelle, to form an autolysosome where the lysosomal enzymes degrade ribosomes, mitochondria, bacteria, viruses, protein aggregates, and all of those particles. Now, ATG9A plays a role in this process. So the small vesicles containing ATG9A derived from the TGN by virtue of AP4 then move to these pre-autophagosomal structures, delivering lipids so that the pre-autophagosomal membrane can expand and eventually close into an autophagosome. And the ATG9A function is required for another autophagy protein named LC3 to convert from an LC3-1 form to an LC3-2 form, indicated in red in this diagram. And we can monitor the integrity of the autophagy pathway by measuring the levels of LC3-1 and LC3-2 by immunoblotting, as shown in this immunoblot. And you can see in the red box that there are two bands. The upper band is LC3-1 and the lower band is LC3-2. An analysis of wild type and AP4 knockout cells shows that there is a decrease in the ratio of LC3-2 to LC3-1, which indicates that there is a defect in autophagy in the AP4 knockout cells. Why? Because ATG9A is stuck at the trans-Golgi network and it cannot get to the forming autophagosomes. Now, in neurons, autophagy is essential for disposing of protein aggregates in certain neurodegenerative disorders, but also under normal conditions that otherwise will cause axonal dysfunction and degeneration. So in our neurons, autophagy is constantly getting rid of abnormal organelles, damaged organelles, and protein aggregates that form. And that disposal is essential for the maintenance of axonal health. If you have mutations in certain proteins and you have an overwhelming amount of aggregates forming in the axon, then that will overwhelm the autophagy machinery, and you will have accumulation of protein aggregates, which will lead to axonal degeneration and death. For example, in Huntington's disease. So to test whether the AP4 knockout neurons had a defect in the disposal of protein aggregates by autophagy, we expressed a pathogenic Huntington disease protein, abbreviated HTT, which forms aggregates indicated by the red arrowheads in the axon of the neurons that you can visualize better on the left. So these are very long, elaborate axons. And so you can see a small number of aggregates indicated in green in the wild-type cells. But when we looked in the AP4 knockout cells, we saw an increase in both the number and the intensity of these Huntington aggregates, indicating that the absence of AP4 results in an autophagy defect, in an inability to dispose of these abnormal protein aggregates in the axon of these neurons. And we think that this is what causes the neuronal pathogenesis, the axonal pathology in both mice and patients with mutations in AP4. So in conclusion, we discovered this complex AP4 many years ago. For many years, we knew a lot of the properties of this complex, but we didn't know what role it played in organismal physiology. We now know that mutations in AP4 cause a disease known as hereditary spastic paraplegia, and that the AP4 knockout mice recapitulate a lot of the symptoms seen in the patients, including thin corpus callosum, axonal dysfunction, and motor deficits. And therefore, the mice are a good model for the human disease, and they are being used in preclinical studies of this disease. And we now know that AP4 mediates signal-dependent export of ATG9 from the TGN. And we know that AP4 knockout cells exhibit autophagy defect, particularly in neurons, and therefore autophagy dysfunction may contribute to the pathogenesis of this particular type of hereditary spastic paraplegia. So in the last couple of minutes, I would like to tell you that these findings have translational implications. In the first place, this cellular phenotype of accumulation of ATG9A at the trans-Golgi network is now being used for diagnosis of this disease. For instance, there may be some patients that have mutations of unknown pathologic significance, so we can now test in fibroblasts for these patients for the accumulation of ATG9A at the TGN, and that is diagnostic for this disease. This phenotype is also being used in drug screens for pharmacologic agents that will be able to remobilize ATG9A from the trans-Golgi network towards the cell periphery as a form of alleviating the autophagy defect and hopefully the defects in the patients. And also this phenotype is being used to monitor gene therapy vectors for use in patients for the ability of the vectors to remobilize ATG9 from the trans-Golgi network. And in this regard, we are collaborating. We are part of a consortium led by Stephen Gray and Xin Chen at the Department of Pediatrics at UT Southwestern Medical School. This is a project that was funded by a charity known as Cure SPG 50 that's led by Terry, who has a son with AP4 deficiency. And as part of this consortium, we tested some of these gene therapy vectors that we found were very effective at remobilizing ATG9A in the upper right picture, where you can see ATG9A accumulated in the TGN of fibroblasts from these patients. And if you look in the lower right picture, you will see that the re-expression of the missing subunit results in dispersal of ATG9A from the transgene network to the periphery. So Michael, on the picture on the left, was the first patient to receive this gene therapy 4 months ago at the Hospital for Sick Children in Toronto upon approval of the clinical trial by the Canadian Regulatory Agency. And now the therapy has been approved by the FDA for clinical trials in the US, which will start very soon. So this has been a long and very fascinating journey to us from the discovery of this complex in 1999 to the elucidation of the function of this complex, to the use of this knowledge now to develop therapies for these patients. And I want to acknowledge the people in my lab over the years who did these studies. They are the real stars of this work. I also want to thank our collaborators in the US and other countries. And also I want to thank my institute, NSCSD, which funded this research for all of these years. And finally, I want to thank all of you for your attention. Thank you very much.
And thank
Susie.
you, Dr. And thank you, Dr. Bonifacino, for that informative presentation. We will now start the live Q&A portion of the webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen. So let's get started.
You will read the questions to me because I don't know if I can see the questions.
Absolutely. And we already have a few questions. Our first question, Dr. Bonifacino, AP4 is expressed in all cells. Why do the patients have only a neurological phenotype?
Yeah, that's a good question. So this is a question that I am asked often when I talk about these neurological disorders of protein trafficking. So I said before that AP4 is ubiquitous. It's present in every cell in our body. So why do the mutations result in a neurological phenotype? And I think the reason is that neurons are much more dependent on protein transport than other cells. If a fibroblast or a keratinocyte or a lymphocyte have a defect in a protein trafficking molecule, they will still manage. They will hobble along. They will still carry out their functions to some extent, and that may not be manifested as a severe defect in patients. But if a neuron has a defect in protein trafficking, that's more likely to result in problems. And that is because the neurons have these very long processes like the axons and the dendrites. Some of these processes may be up to 1 meter long. For instance, the neurons that innervate your toe so that you can move your toe, they have axons that are up to 1 meter long. The body of the neuron is in the spinal cord, but the tip of the axon that's moving the muscle is 1 meter away. So if you have a problem with moving proteins in the neurons, they will be manifested much more severely in neurons than in other cell types. And so that's a common finding in a lot of diseases involving components of the protein trafficking machinery. So I hope that answered the question.
Absolutely. Thank you so much. We have one more question coming in. Do all coagulopathies have a neurological presentation?
Yeah, that's related to the first question. So coagulopathies, this is a term that we coined recently because mutations in AP1, AP2, AP3, AP4, AP5, and many other code proteins that I didn't mention in my talk, they cause genetic disorders that we refer to as coagulopathies. All of these protein codes are components of the protein trafficking machinery. And for the same reason that AP4 mutations cause neurological disorders, mutations in all the other AP complexes and other code proteins also cause neurological disorders. There are many of these disorders now, dozens of these disorders, and most of them manifest neurological symptoms. And the reason being that protein trafficking is really essential, more essential in neurons than it is in other cells.
Thank you so much. And one more question, Bonifacino. What are the next steps that you will be taking in your lab to study?
Yeah, so the AP4 deficiency is only one of several neurodevelopmental disorders that we study in our lab. And our interest is elucidating the mechanisms of these disorders in the hopes that we can come up with therapies. Because if we don't know how mutations in these genes cause disease, what the mechanisms of disease are, it's unlikely that we will be able to come up with new therapies for these disorders. And so a major focus of our lab is to understand the mechanisms of disease so that we can come up with therapies, with pharmacological interventions or gene therapy interventions so that we can treat these children. It is estimated that about 1 to 2 newborns in the US will develop intellectual disability. And that's only one of various neurological disorders. So although each of these diseases is rare, when you combine all of the movement disorders and the intellectual disability disorders, they affect a significant proportion of the population. So my lab, as well as many other labs, are working to try to come up with new therapies for these disorders through the understanding of the molecular mechanisms that underlie the diseases.
Thank you so much, and such an incredibly important mission. Dr. Bonifacino, thank you for your time today. Would you like to provide any closing remarks for our audience before we go?
Well, yeah, I'm not sure exactly who my audience is, but I just want to mention something. Thank again my institute, NICHD, which is one of the about 28 institutes that make up the NIH for funding this research. NICHD was established 60 years ago. This year is the 60th anniversary of our institute. And it was established during the Kennedy administration through the advocacy efforts of the president's own sister, Eunice Kennedy Shriver, who lobbied the government to establish an institute whose mission would be to combat mental retardation, as it was known at the time, intellectual disability, as we refer to these days. And so that was the initial mission of NICHD, and it remains a central mission these days. And within a few years, within the first decade of NICHD's existence, a major cause of acquired intellectual disability was addressed through the development of the vaccine to Haemophilus influenza, also known as meningococcus, which before that vaccine caused intellectual disability in many children in the US and around the world. And after that vaccine started to be used in children, and anybody who has children will know that this is a vaccine that children receive within the first year of age. And that was eradicated as a cause of intellectual disability in the US and in many other countries throughout the world. So that really shows how investment in basic and translational research leads to actionable treatments for children. And we hope that we can replicate that success for other types of intellectual disability and other diseases in children.
Well, Dr. Bonifacino, thank you again for your time today and for this very important research. And before we go, I want to thank our audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted on demand will be addressed by our speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until September 21st, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care. Be safe, everyone. Bye-bye.
Thank you.